Thanks for the introduction. So I'll be talking about mRNA translation today. Does it work? It's not connected.
Is it on? Okay.
Oh, yes. Right, thank you. Okay, so I'll be talking about mRNA translation. The rate limiting step of the protein biosynthesis is translation initiation. It can be achieved in two ways, either cap dependently or cap independently. Most of the cellular mRNAs, as well as the mRNAs, from the mRNA vaccines, are translated cap dependently. In this mode of translation, the 5' cap that's at the 5' end of the mRNA is recognized by EIF or F complex, and then it leads to the ribosome recruitment. It works very well in normal conditions. However, under cellular stress, the assembly of the EIF or F complex can be inhibited, which leads to attenuated ribosome recruitment. This creates a challenge if you want to design therapeutics mRNA to target tissues under cellular stress. So for example, if you want to develop mRNA therapeutics against cancer by aiming to express tumor suppressors, cytokines, or maybe Cas9 for gene editing. So potential solution to this could be the use of cap-independent translation. In this mode of translation, the ribosomes can be recruited through IRES, which is internal ribosome entry site, a secondary structure on the mRNA that enables binding to translation factors and subsequent recruitment of ribosomes. So cap is not required in this mode of translation. This mode of translation is resistant to translation inhibition under cellular stress. However, in general, the cap-independently translated mRNAs are translated less efficiently than capped mRNAs. This is for many reasons, but most importantly, they are less stable than capped mRNAs because of unprotected 5' end, as well as the methods for characterization and development of cap-independently translated mRNAs are limited. So we aim to improve the properties of the cap-independently translated mRNAs, and we look at the 5' end. We took the inspiration from the way how the 5' cap is introduced to capped mRNAs. So reagent, like for example, CleanCap AG, developed by TriLink and used in mRNA vaccines, can be added to the mixture of NTPs and used in the in vitro transcription to efficiently prime the transcript. So this reagent will be incorporated at the 5' end of the mRNA. This process has remarkable efficiency, around 95% or more of the transcripts will be successfully primed with this reagent. So we used a similar reagent, but instead of the part that's interacting with EIF or E protein, we used azides that enabled modification of the mRNA using click chemistry. So this reagent can be similarly as CleanCap very efficiently incorporated into the mRNA. As you can see here, it works very well at broad range of concentrations. And the mRNA that incorporates this reagent can be very easily post-transcriptionally modified using strain-promoted azide alkyne cycloaddition. Just in 1 hour, we can achieve a complete conversion. So we started from examining translational properties of the modified mRNA. As a model, we used mRNA with IRES sequence released from HCV virus and EGFP as a reporter, and we used flow cytometry to assess the translational properties. And as you can see here, the version with the dye incorporated at the 5' end exceeded a significantly higher translation efficiency than the triphosphate version. It's around 8-fold increase in translation efficiency, while the azide version actually showed slightly decreased translation efficiency. So we took a further look at other properties of the mRNAs, like cellular stability and immunogenicity, to find out that the version with the dye of the 5' end was more stable than the 5' transphosphate, but actually the 5' azide was less stable. This is because 5' triphosphate mRNA, in order to be degraded, has to first undergo hydrolysis of the triphosphate to monophosphate, and then XRN1 enzyme can degrade the mRNA from 5' to 3' end. However, in the case of azide, probably can be recognized directly by the XRN1 enzyme to trigger degradation. Another element contributing to the translation of the mRNA is its immunogenicity, actually. So 5' triphosphate mRNAs are frequently occurring in viruses, and they're recognized by cell as viral RNAs and trigger immune response. This is through RIG1 receptor. And you can see that for the version of the mRNA that has terminal 5' triphosphate, we could see elevated expression of viral infection-related cytokines in mature dendritic cells. But introducing modification, either dye or azide, solves the problem, and you can see a decrease of the expression of the cytokines. So I mentioned before that the characterization of the independently translated mRNAs is challenging. Currently, the approach that's most commonly used for characterization of these mRNAs is dual reporter bisystronic assay. In this strategy, there is mRNA that contains two C-strands. First C-strand that serves as a reference is translated cap dependently, while the second C-strand, the one that you're actually interested in, that will inform about the efficiency of iris authentication, can give expression of some other reporter protein. So we can use the first C-strand to normalize the expression of the reporter of interest to rule out, for example, differences in the transfection efficiency. However, if we want to use this strategy to assess the efficiency of cap-independent translation between different cell lines, it is challenging because the expression of translation factors in different cell lines can vary. So we cannot ensure that the expression of the first C-strand will be similarly efficient. So as an alternative to the strategy, we propose to use the dye of the 5' mRNA to normalize the expression of the reporter protein to the total amount of the mRNA delivered. So to test the strategy, we looked at 3 different irises, 2 derived from viruses and 1 modified that we call ISIN or synthetic iris. And we tested the translational properties of those constructs in for cell lines. And just to give you the idea of the normalization concept, we can see that the raw expression of the reporter protein in HeLa cells is higher than in HEC293 cells for the IHCV construct, but if we look at the amount of the mRNA delivered, it looks also similar. That's more mRNA is delivered to the HeLa cells compared to HEC cells. So if we normalize the expression of the protein to the amount of the mRNA delivered, we can see that actually the mRNA productivity, so the amount of protein that's made from the same amount of mRNA, is the same for the 2 cell lines. So as we can track the amount of the mRNA delivered to the cell, we can also very easily derive those response curves. And we don't have to do many transfections at different concentrations of the mRNA. We can just literally use data from one well because we are looking at single cells in a large population. So we'll have cells not transfected or transfected with a large amount of mRNA and everything in between. So we can very easily confirm the validity of the assay by looking at if the translation machinery of the cell is not saturated. So from the benchmark, we could see that the Eisen construct was performing the best. So we're curious, how will it compare to some established constructs, like the starting construct that's translated cap independently that I presented on the previous slide, as well as some established constructs like cap-dependently translated linear mRNA, as well as circular RNA. And the modified construct with engineered sequence called Eisen was performing 67.1 fold better than the starting construct that was cap-independently translated. And the modified construct was performing similarly well as capped and circular mRNA. This was actually surprising to us, especially if we compared it to circular RNA. So we additionally confirmed the circularity of the RNA and the linearity of our construct by performing RNAs are digestions. As you can see from this data, the linear version was degraded from 3' to 5', and while the circular, because it doesn't have 3 ends, didn't undergo the degradation. So finally, we explored the possibility of applying the strategy for cellular phase tracking of the mRNA. And as you can see from this data, we can very easily track the uptake as well as the expression of the protein. We can either use flow cytometry or we can use microscopy to, for example, see what's the lag between the delivery of the mRNA and the expression. Furthermore, we wanted to see how it performs in some more demanding applications. So we performed time-lapse imaging as well as live cell 3D imaging that are more demanding because there is much more light that is, let's say, delivered to the cell. So it's more, let's say, the constructs can be more prone to photobleaching. And as you can see here, we can very nicely track the delivery of the mRNA to the cell, as well as, thanks to very strong expression of the protein, we can resolve such fine details as the retraction fibers in the mitotic cell. So you can ask, well, if the signal is actually corresponding to the localization of the mRNA or maybe free die. So to confirm that the localization indicates the actual localization of the mRNA, we performed FISH, and we correlated the signal of the probe attached to the mRNA with the FISH probes. And as you can see here, the correlation was really good at 6, 12, and 24 hours, despite the degradation of the mRNA release of free dye. We think it's because the dye doesn't relocate to other components and stays at the same place where it was originally. So to sum up, we developed a toolkit for improvement of properties of cap-independent fecal-sensitive mRNAs that is not only by direct improving stability and translation, but also by facilitating detailed characterization of the cap-independent fecal-sensitive mRNAs. I want to thank my supervisor, Tom Brown, as well as co-workers also involved in this project, Brendan and Ahab, and thanks for your attention.